Genome-wide association studies, or GWAS, produce a set of genomic loci where there are statistically significant associations between specific sequence variants and the risk of a disease or magnitude of a trait. But the ultimate goal of this research is to identify which genes at these loci are involved in disease risk. Often, after performing a GWAS, researchers will combine multiple other data types and methods in order to predict the causal or effector gene at each locus. Usually these predictions and their supporting evidence are presented in supplementary data files, where they are static and relatively inaccessible. We believe these lists are very useful because they give a consensus view of the genes most likely to be important for a disease or trait. So we're curating them from the literature and presenting them in the knowledge portals. This video will illustrate the effector lists in the Common Metabolic Diseases Knowledge Portal, but it's applicable to all the knowledge portals. To see lists of predicted effectors, click the Tools menu and go to the Predicted Effector Genes link. Now, to see which lists are available, click on the Selection menu. The lists that are available in each portal are those that predict effector genes for the diseases and traits that are the focus of that portal. To access a list, click on one of the names in the menu, and then click on the blue button with the phenotype. The top of the effector list page shows the reference for the list and gives a brief summary of how it was generated. Scrolling down, the table of predictions is located on the View Data tab, while the Research Method tab gives more details about the method. The options directly above the table allow you to set the number of rows displayed, to save the table in CSV or JSON format, and to choose which columns you would like to display. You can sort the table by clicking on a column header. Some column headers also display additional information on mouse over. Click the View button to see all the supporting evidence for a row. Above the table, the filters allow you to find results of interest. For example, start typing a text string and hit return. We typed CS and now we see the gene names that contain CS. You can do the same kind of search with RSID, typing letters and numbers. Other filters, like the priority score filter here, allow you to set an upper or lower threshold for numerical values. Here we'll put in 8, and now we see only the genes with priority score of 8. Finally, for more written documentation, you can go to the Help menu, click on Tools, and click on Predicted Effector Genes. We're very interested in displaying predicted effector lists for a wide range of diseases and traits. If you have generated a list and would like to display it on the knowledge portals, please contact us.